This is Mrs. Palmer-Quay, and this is the second video for Unit 2 of Advanced Biology. In this video, I want to talk to you briefly about the macromolecules that are very important for sustaining life. So macromolecules are organic molecules, and organic molecules are carbon-based molecules. And most of a cell, our cells or bacteria cells in this particular example that I was able to find, are made up of water. Here we've got 70%. We are 65 to 70% water ourselves. And of the rest of it, about 4% are small ions, other small molecules, inorganic compounds. But 26% is the macromolecules, the organic compounds. And of that, we have four classes that we're going to be talking about. Polysaccharides, which are carbohydrates, proteins, DNA and RNA, which are nucleic acids, and then phospholipids representing the lipid group. So carbon can form four bonds. If you remember from chemistry, carbon has four valence electrons, so it can bond to four different atoms in a single bond, or it can form double bonds or triple bonds. Carbon, therefore, can form a variety of shapes, a variety of skeletons, such as the straight lines we see here, all single bonds, or a branched shape, again, all single bonds, or here is a straight line with some double bonds, or even a ring. So because these different shapes are possible with carbon, there are many functions that the carbon-based molecules can have in an organism because they can take on a variety of shapes. When we're talking these macromolecules, these large carbon-based molecules, they're also known as polymers. And polymers are built from smaller molecules called monomers. So many monomers are put together to form a polymer, and then all together these form the macromolecules that we're talking about today. So we have four classes of macromolecules. The first one, proteins. The monomer is amino acids. Lipids is a second class, and they aren't exactly made up of monomers like proteins, but they all do contain at least one fatty acid chain. So that is a unit that repeats among the different lipids. Carbohydrates are another macromolecule, and monosaccharides or simple sugars are the monomers of the carbohydrates. And then nucleic acids are the fourth class of macromolecules, and the nucleotide is the monomer in this situation. Monomers are linked by a process called condensation or dehydration synthesis, and of course this word dehydration should clue you into the fact that we're dealing with water. So really removing a molecule of water. And if you look here, we've got two monosaccharides that we're going to join together to make a disaccharide by taking the hydroxyl group off of this particular end of this molecule and just the hydrogen out of this. And if you remove those two things, you're going to be making water, and the oxygen then becomes a bridge in the middle. So these are now linked together. So this is how dehydration synthesis works, by removing a molecule of water. The opposite can happen if you want to break down a polymer. Instead of taking water out, you put water in. So if you put water into this bond, then the oxygen will break here and it'll pick up a hydrogen, and in this space we'll add the hydroxide. And so then you have again two separate monomers of a monosaccharide instead of a joined molecule. So hydrolysis is what we call adding water to break things apart, whereas dehydration synthesis is taking away water to put molecules together. So I'm going to start with carbohydrates. Carbohydrates are some things that are common to them. They all contain carbon, hydrogen, and oxygen, and they're usually in the proportion that we could write CH2O in general. There are small mono and disaccharide molecules that we know because they taste sweet. Slightly larger, we call them oligosaccharide molecules, serve as antigens on cells. If you remember, these are markers that say this cell belongs to me or this cell does not belong to me. So the slightly larger carbohydrates make up those antigens, and then very large polysaccharides can provide energy storage or structure. They can be straight or slightly branched, and we'll get to specific examples in a minute. So monosaccharides are called simple sugars, and they are things like glucose and fructose and galactose. And if you notice here, here's an example of these three sugars, and they all would have the structural formula of C6H12O6. But they look slightly different. In this case, if you look at the hydroxide and hydrogen position here, you see it is reversed between glucose and galactose. And remember, these are actually 3D molecules that we are trying to draw on a flat surface, and so that these hydroxide and hydrogens would be sticking out at different angles. And in the case of fructose, we see the double bond to the oxygen is on a slightly different carbon in the chains position than it is on the other two. So these are known as isomers, and since isomers change the physical structure slightly, you do have different properties with different isomers. Even though the chemical formula is exactly the same, they behave differently or they're taken up by the body slightly differently because their structure is different. You may notice again all these things end in ose, galactose, fructose, glucose, and this ose ending tells you that it's a sugar. These monosaccharides can also change, they can switch back and forth from this chain structure to a ring structure. This especially happens in aqueous solutions, and of course you are 65 to 70% water, so you are basically in aqueous solution. So often the glucose is found in a ring structure when it's inside your body. And this is just an example of the shorthand that scientists like to use. We get tired of writing all these hydroxides and hydrogens out on the carbons, and so we'll just draw a straight line, and it's assumed that when we're talking about a sugar, there's going to be a hydroxide and a hydrogen attached to them, and there's a carbon at all of these corners. So disaccharides, di means two, so disaccharide is made by joining two monosaccharides. Again, we're looking at taking the hydroxide off of one side and the hydrogen off of the other. These come together to give us water. It is a waste product of this reaction, and then this oxygen then becomes a bridge between the two molecules. We call this a glycosidic bond. Common disaccharides include sucrose, which is white table sugar, and it is a combination of glucose and fructose bonded together. Lactose, which is glucose and galactose. This is milk sugar. Many people have a reaction to lactose and it gives them stomach ache. And maltose, which is a grain sugar, and it is two glucose molecules put together. Polysaccharides are long chains, sometimes basically just straight chains, sometimes with small branches, sometimes with some other linkages going on. And these long chains of monosaccharides, or long chains of glucose actually, have different properties. The starch, this long single chain here is how extra glucose is stored in plants. So we've got a farmer here with some potatoes, and the potatoes, of course, we know are a starchy vegetable. And so the plant stores extra glucose as starch, and we, of course, can then eat the starch and release these glucose molecules, digest them off, and use them for energy. In animals, we store extra glucose again as glycogen. So this is the animal form of storage polysaccharide. The equivalent to starch is in plants, glycogen is in animals. And then cellulose is a polysaccharide that provides a great deal of structure. It's very tough. And even though it's made up of individual glucoses, it has a different direction in the bond between them, and our enzymes are not able to fit with the shape of the bond between the glucose and actually digest this. That's why cellulose cannot be digested by animals. The animals that eat cellulose plants, the ruminants, are able to do that because they have lots of bacteria in their guts that are actually doing the digestion for them. The next class of macromolecules I want to talk about are lipids, and they're not exactly polymers, but they all have at least one fatty acid chain. Because they have a fatty acid, or we see the fat here, and we know that oil and water doesn't mix. So most lipids are hydrophobic. They are water-fearing. They don't dissolve in water. They don't mix with water. And so lipids include fats and waxes and steroids and oils. And this is just an illustration of a fat molecule. We have two general types of fatty acids, saturated and unsaturated. Saturated fatty acids are full of hydrogen. There are no double bonds or triple bonds. Everything is a single bond between carbon and hydrogen down the line. But in an unsaturated fatty acid, there is a double bond. There may be more than one double bond, so that you've got a place where hydrogen has been taken out or hydrogen can actually be put in. And this is what happens when we partially hydrogenate vegetable oils and turn them into things like vegetable shortening. We are adding hydrogen and breaking this double bond, turning it back into a single bond and putting in more hydrogens. A common type of lipid is the triglyceride, which is what makes up all of the dietary fat that we enjoy in so much food. And triglycerides serve as energy storage. They provide calories, more calories than any other of the types of macromolecules that we eat. And on our bodies, they provide some insulation and protection around organs like our eyes or our kidneys. Fatty acids are composed of a glycerol backbone. This is an organic alcohol, a 3-carbon chain with hydroxide groups on one side and hydrogen every place else, and fatty acid chains that connect to this glycerol. So here's an example of a fatty acid chain that has been connected again by dehydration synthesis. Water has been taken out, hydroxide group off of one carbon and the hydrogen off the other side. Oxygen will then serve as a bridge between the glycerol and the fatty acid. And this forms, essentially it's the same process, but because we're dealing with lipids, we call it an ester bond instead of a glycosidic bond. And so this is one fatty acid chain attached, and there'd be a second one here and a third one when we were all done, because this is a triglyceride. So in that, there would be 3 fatty acid chains attached to that glycerol backbone. Lipids have a very important role in cell membranes. The lipids there are not triglycerides, though they are phospholipids. So they do have 2 fatty acid chains attached to that glycerol, but the third one is taken up by a phosphate group. And so the phosphate group is a water-loving, a hydrophilic group. It's polar. It will be attracted to water. But the lipid ends, as you know, water and fat don't mix, so they are hydrophobic or nonpolar, not attracted to water. So if you remember all these diagrams of the cell membranes, we would draw these little phospholipids with their lipid tails to the inside and the phosphate heads to the outside, because the watery environment outside the cell is here and the watery environment inside the cell is here, and those will be attracting the phosphate groups, whereas in the middle you have the hydrophobic lipid tails. Another important role of lipids is in the formation of steroids, and the base molecule for steroid is cholesterol. Cholesterol had a very bad name for many years in the medical world, but it's very important in your body. It's essential to health. You can make all the cholesterol you need, so you don't really need to eat any. But it has a fatty acid chain, not particularly long, but here's one. So there's a carbon at all of these little corners. And it has 4 rings that are fused together. And from cholesterol, you can make a variety of other steroids. You can make these sex steroids, testosterone and estrogen. It also is linked inside those phospholipids that we have in the cell membrane. It helps keep them, keep their right places, especially as temperature changes are experienced by the cell. So it has a lot of very important roles, but you don't need to eat any. Your body can make it for you. Proteins, our third class of macromolecules, they're put together by amino acids. Long chains of amino acids make proteins. There are 20 different amino acids, but you really only need to eat 7. There are 7 essential amino acids. The rest of them you can make in your body, but 7 of them you have to get from food. So if you want to be a vegetarian, that's why vegetarians or vegans eat things like beans and rice, because together they give you the right combination of essential amino acids. Proteins have a variety of roles in our bodies. You really are most of your body is protein, besides the part that is water. So it is used to be parts of your cells, and it's part of these hormones and enzymes especially. It's part of muscles to facilitate movement. Proteins really do much of the work of the cell, because proteins are much of the cell once you take out the water. An amino acid all have a basic structure. You've got a central carbon. On one side it's attached to a carboxyl group, this COOH group. And on the other side you have an NH2 or an amine or amino group. And then on the other end of the carbon you have a variety of things, and that's just generally known as the R group. And it's the particular characteristics of whatever other atoms are attached here on the R group position that determine what the amino acid is like. So for example, we have two amino acids down here, leucine, and it has this particular side group, and this side group is hydrophobic. So it would not like to be near water. It would want to be far away from the watery environment of the cell. But serine, this small side group for this amino acid, is hydrophilic. So it is water-loving. So the amino acids are what determine the properties of the amino acid side groups determine the properties of the amino acids, and then all of the amino acids put together determine the property of the protein. Cells link amino acids by dehydration synthesis again, so we take that water out, and here instead of oxygen providing a bridge, we have a bond between carbon and nitrogen, and these are called peptide bonds when we come to proteins. So each particular macromolecule has its own particular bond, but the process on how we get to that bond is essentially the same. Now, proteins are made up of hundreds of amino acids, and they don't just exist as one long chain. They exist as blobs. They're bent and curled and twirled. And so we talk about protein having 3 or possibly 4 different structures. The primary structure is provided by that amino acid chain. The order of the amino acids, we've got valine and glycine and serine and leucine here, that provides the primary structure. But because there are different combinations in the R groups, those R groups may cause some hydrogen bonds to form between hydrogen and nitrogen or hydrogen and oxygen, and so it can cause that long chain to start to twirl up here in this alpha helix, or maybe it bends back on itself and sort of folds a little bit as it moves. It's called the pleated sheet. And so these first twirls and bending back on itself are known as a secondary structure caused by hydrogen bonds, and then the amino acids that are all hydrophilic might group together, or the ones that are all hydrophobic might group together, or ones that are a little more acid or a little more basic. And so this forms the tertiary structure. So one protein chain will go up through a tertiary structure to form some kind of blob, and you can find these interesting pictures that kind of look like this. But in some proteins, it's actually 2 protein chains that work together or more. And so here you have quaternary structure, or 4th level of structure on how those 2 proteins work together. So proteins are really quite complex organisms, and this folding and bending shape is what makes them able to do what they do, especially as enzymes. Anything that changes that shape will change their ability to do their job. Sickle cell anemia or sickle cell disease is an example of a change in primary structure causing an ultimate change in the protein's ability to do a function. The normal hemoglobin here is 146 amino acids long, and if one mistake, valine gets substituted for a glucine, just one amino acid, you see the hemoglobin changing its shape to the extent that the normal red blood cell that looks sort of a donut shape with the middle still filled in turns into this sickled shape, sort of pointy on the ends, not fat and plump. It loses, in the sickle shape, it loses its ability to carry oxygen as well. The pointed ends get stuck in blood vessels and cause blood clots, which then can lead to pain. Can be a very debilitating disease. It's a genetic disease because the directions for hemoglobin are incorrect. So it's just one tiny little amino acid out of 146, but it has an impact all the way down through those different levels of structure and ultimately then the function of the protein. Proteins are also essential as enzymes. Enzymes are biological catalysts. They speed up reactions because they lower the amount of activation energy needed to get the reaction going. Without enzymes, we wouldn't be alive because we wouldn't have these reactions happening in our body, and there are thousands of different enzymes. Enzymes are big blobby proteins again. They have a particular place on their protein known as the active site that has a particular shape that matches whatever chemical reaction this enzyme is going to be speeding up. The chemicals that are involved in that are known as the substrate, and the substrate fits perfectly into the active site. Then the position of the enzyme, the position that the enzyme holds this substrate, then allows the reaction to happen, and once that's done, the substrate, which has been changed, is released and the enzyme is ready to move on. Enzymes are specific for one type of substrate, and anything that changes the shape of this enzyme will change the shape of the active site and could potentially render it inefficient, unable to function at all. Some key proteins. Insulin is a protein that you may have heard of. It causes the liver to take up excess sugar that's found in the bloodstream and put it into that polysaccharide glycogen, the animal form of storage for carbohydrates. Cell membranes also contain many different types of proteins, and one important role they play is as receptor proteins, which help cells communicate and recognize each other. Your cells are actually talking to each other in your body. And then the final class of macromolecules are nucleic acids, and they have a critical role in making all of the body's proteins. DNA is the library of genetic information, given the blueprints that are needed to make all of those proteins, and RNA is the transcriber and translator of the genetic information into the proteins. They work together. DNA stays in the nucleus, RNA travels out to the cytoplasm, and both of them are nucleic acid molecules. Nucleic acid macromolecules are polymers of nucleotides, and like the amino acids, these have a particular structure. They have a sugar. In the case of DNA, it's deoxyribose. In the case of RNA, it's just plain old ribose, which is attached, the sugar is attached to a phosphate group. And then this is always the same. You always have the same sugar and the same phosphate group on DNA or RNA, but these nitrogenous bases will differ. There are 4 possibilities, and we use letters to identify them, so it can be adenine A, guanine G, thymine T, cytosine C. Thymine is only for DNA. In RNA, uracil substitutes for that, but there's still only 4 base possibilities. And so these are put together that the sugar and the phosphate here, the blue and the yellow, they form the backbone of the molecule, and the bases are sticking out off that backbone. In DNA, these 2 strands actually join together, and they're held together in here by hydrogen bonds. Remember, hydrogen bonds do not share an electron, but there is a strong attraction between because of the chemical structure of these 2 nitrogenous bases. This double helix is able to hold together. RNA, unlike DNA, is only a single strand. RNA and DNA work together, as I said, to make proteins. We have a DNA in the nucleus, and the messenger RNA transcribes the information off of the gene, travels out to the cytoplasm, and hooks up with transfer RNA and the amino acids that these tRNA molecules pick up from the cytoplasm. You have the codon here, these 3 base sequences that match up, and then you have a protein being built. I'm not going to say any more about this process, but I will give you information on another video that you can watch, not done by me, but which will give you more detail if you don't remember all of this. Another important nucleotide is ATP, or adenosine triphosphate. And ATP is used by cells for energy. They're kind of like little energy batteries. They're made up of a nucleotide that has 3 phosphate groups instead of just 1. So here's your ribose, here's your sugar. This is a different kind of molecule drawing. And then we have attached to that adenosine, we have the nitrogenous base, and then 3 phosphates, not just 1, but 3 phosphates group. And energy is kept in these bonds. When the bonds are broken between the phosphate group, this is when energy comes out and can be grabbed by the cell to help power all of the cell's processes. So this is another nucleotide. We sometimes don't think of it as being part of this group, but it is. So that finishes the quick overview of macromolecules. For the most part, you are what you eat because we are consumers, not producers, and so we have to take in our food. We take them in as macromolecules and then digest them into their individual monomers and then reassemble the food particles into the macromolecules that we need in our body. So this phrase, you are what you eat, is truer than you know. So hopefully that'll give you a little sense of some of the important things that you need to know about these different molecules as you work on this unit.